Genetics is the study of biologic inheritance, and understanding of genetics is essential to study human, animal, plant, and microbial life. In the 19th century, microscopic studies of cells led scientists to suspect the nucleus of the cell contained the important mechanisms of inheritance. Scientists found chromatin, the substance giving the nucleus and granular appearance, is observable in non-living cells. Just before the cell divides, the chromatin condenses to form discrete, dark staining organelles, which are called chromosomes. With the rediscovery of Mendel's important breeding experiments at the turn of the century, it soon became apparent that chromosomes contained genes, the basic units of inheritance. The primary constituent of chromatin is deoxyribonucleic acid, DNA. Genes are composed of sequences of DNA. By serving as the blueprints of proteins in the body, genes ultimately influence all aspects of body structure and function. Humans have approximately 20,000 protein coding genes and at least an additional 9,000 to 10,000 genes that encode various types of ribonucleic acid RNA. They are not translated into proteins. An error in one of these genes often leads to a recognizable genetic disorder. To date, more than 20,000 genetic traits and diseases have been identified and cataloged. As infectious diseases continue to be more effectively controlled, the proportion of beds in pediatric hospitals occupied by children with genetic diseases has risen. In addition to genetic diseases in children, many common diseases primarily affecting adults, such as hypertension, coronary heart disease, diabetes, and cancer, are now known to have important genetic components. Great progress is being made in the diagnosis of genetic diseases and in the understanding of genetic mechanisms underlying them. With the huge strides being made in molecular genetics, genotherapy, the utilization of normal genes to correct genetic, has begun. Composition and structure of DNA. Genes are composed of DNA, which has three basic components: the 5-carbon monosaccharide deoxyribose, a phosphate molecule, and 4 types of nitrogenous bases. 2 of the bases, cytosine and thymine, are single carbon nitrogen rings called pyrimides. The other 2 bases, adenosine and guanine, are double carbon nitrogen rings called purines. The 4 bases are commonly represented by their first letters: A, adenine; C, cytosine; T, thymine; and G, guanine. Watson and Crick demonstrated how these molecules are physically assembled as DNA, proposing the double helix model, in which DNA appears like a twisted ladder with chemical bonds as its rungs. The 2 sides of the ladder consist of deoxyribose and phosphate molecules. United by strong phosphodiester bonds, projecting from each side of the ladder at regular intervals are nitrogenous bases. The base projecting from one side is bound to the base projecting from the other by a weak hydrogen bond. Therefore, The nitrogenous bases form the rungs of the ladder. Adenine pairs with thymine, and guanine pairs with cytosine. Each DNA subunit consisting of one deoxyribose molecule, one phosphate group, and one base is called a nucleotide. DNA directs the synthesis of all body's proteins. Proteins are composed of one or more polypeptides, which in turn consist of sequences of amino acids. The body contains 20 different types of amino acids. They are specified by the four nitrogenous bases. To specify 20 different amino acids with only four bases, different combinations of bases occurring in groups of three are used. These triplets of bases are known as codons. Each codon specifies a single amino acid in a corresponding protein. Because there are 64 possible codons but only 20 amino acids, there are many cases in which several codons correspond to the same amino acid. The genetic code is universal. All living organisms use precisely the same DNA codes to specify proteins, except for mitochondria. The cytoplasmic organelles in which cellular respiration takes place. They have their own extranuclear DNA. Several codons of mitochondrial DNA encode different amino acids compared with the same nuclear DNA codons. DNA replication consists of breaking the weak hydrogen bonds between the bases, leaving a single strand with each base unpaired. The consistent pairing of adenine with thymine and guanine with cytosine, known as complementary base pairing, is the key to accurate replication. The unpaired base attracts a free nucleotide only if the nucleotide has the proper complementary base. When replication is complete, a new double-stranded molecule identical to the original is formed. The single strand is said to be a template or a molecule on which a complementary molecule is built and is the basis for synthesizing the new double strand. Several different proteins are involved in DNA replication. The most important of these proteins is an enzyme known as DNA polymerase. This enzyme travels along the single DNA strand, adding the correct nucleotides to the free end of the new strand and checking to ensure that its base is actually complementary to the template base. This mechanism of DNA proofreading substantially enhances the accuracy of DNA replication. A mutation is any alteration of genetic material. One type of mutation is the base pair substitution, in which one base pair replaces another. This replacement can result in a change in the amino acid sequence. However, because of the redundancy of the genetic code, many of these mutations do not change the amino acid sequence and thus have no consequence. Such mutations are called silent mutations. Base pair substitutions altering amino acids consist of two types: missense mutations, which produce a change, i.e., the sense, in a single amino acid, and nonsense mutations, which produce one of three stop codons. The messenger RNA missense mutations produce a single amino acid change, whereas nonsense mutations produce a premature stop codon in the mRNA. Stop codons terminate translation of the polypeptide. The frameshift mutation involves the insertion or deletion of one or more base pairs of the DNA molecule. These mutations change the entire reading frame of the DNA sequence. Because the deletion or insertion is not a multiple of 3 base pairs, which is the number of base pairs in a codon, frameshift mutations can thus greatly alter the amino acid sequence. Agents known as mutagens increase the frequency of mutations, and examples include radiation and chemicals such as nitrogen mustard, vinyl chloride, alkylating agents, formaldehyde, and sodium nitrate. Mutations are rare events. The rate of spontaneous mutations in humans is about 10 to the 4th power to 10 to the 7th power per gene per generation. This rate varies from one gene to another. Some DNA sequences have particularly high mutation rates and are known as mutational hotspots. DNA is formed and replicated in the cell nucleus, but protein synthesis takes place in the cytoplasm. The DNA code is transported from nucleus to cytoplasm, and subsequent protein is found through 2 basic processes: transcription and translation. These processes are mediated by RNA, which is chemically similar to DNA, except the sugar molecule in ribose rather than deoxyribose and uracil rather than thymine is one of the 4 bases. The other bases of RNA, as in DNA, are adenine, cytosine, and guanine. Uracil is structurally similar to thymine, so it can pair with adenine. DNA usually occurs as a double strand, whereas RNA usually occurs as a single strand. In transcription, RNA is synthesized from DNA template, forming messenger RNA, which is mRNA. A RNA polymerase binds to promoter site, a sequence of DNA that specifies the beginning of a gene. RNA polymerase then separates a portion of the DNA, exposing unattached DNA bases. One DNA strand then provides the template for the sequence of mRNA nucleotides. The sequence of bases in the mRNA is thus complementary to the template strand. And except for the presence of uracil instead of thymine, the mRNA sequence is identified as identical to that of the other DNA strand. Transcription continues until a termination sequence. Codons that act as signals for the termination of protein synthesis is reached. Then the RNA polymerase detaches from the DNA, and the transcribed mRNA is free to move out of the nucleus and into the cytoplasm. When the mRNA is first transcribed from the DNA template, it reflects exactly the base sequence of the DNA. In eukaryotes, many RNA sequences are removed by nuclear enzymes, and the remaining sequences are spliced together to form the functional mRNA that migrates to the cytoplasm. The excised sequences are called introns, and the sequences that are left to code for proteins are called exons. In translation, RNA directs the synthesis of polypeptide interacting with transfer RNA. A clover leaf-shaped strand of about 80 nucleotides. The tRNA molecule has a site where an amino acid attaches. The 3-nucleotide sequence at the opposite side of the clover leaf is called the anticodon. It undergoes complementary base pairing with an appropriate codon in the mRNA, which specifies the sequence of amino acids through tRNA. The site of actual protein synthesis is the ribosome, which consists of approximately equal parts of a protein and RNA. During translation, the ribosome first binds to an initiation site on the mRNA sequence and then binds to its surface so that base pairing can occur between tRNA and mRNA. The ribosome then moves along the mRNA sequence, processing each codon and translating an amino acid by way of the interaction of mRNA and tRNA. The ribosome provides an enzyme that catalyzes the formation of covalent peptide bonds between the adjacent amino acids, resulting in a growing polypeptide. When the ribosome arrives at a termination signal on the mRNA sequence, translation and polypeptide formation cease. The mRNA ribosome and polypeptide separate from one another, and the polypeptide is released into the cytoplasm to perform its required function. Human cells can be categorized into gametes, which are sperm and egg cells, and somatic cells, which include all cells other than gametes. Each somatic cell nucleus has 46 chromosomes in 23 pairs. These are diploid cells, and the individual's father and mother each donate 1 chromosome per pair. New somatic cells are formed through mitosis and cytokinesis. Gametes are haploid cells. They only have 1 member of each chromosome pair for a total of 23 chromosomes. Haploid cells are formed from diploid cells by meiosis. In 22 of the 23 chromosome pairs, the 2 members of each pair are virtually identical in microscopic appearance, thus they are homologous. These 22 chromosome pairs are homologous in both males and females and are termed autosomes. The remaining pair of chromosomes is the sex chromosomes. They consist of 2 homologous X chromosomes in females and a non-homologous pair X and Y in males. A metaphase spread, which is a photograph of the chromosomes as they appear in the nucleus of somatic cells during metaphase. In figure 210A, the chromosomes are arranged according to size with the homologous chromosomes paired. The 22 autosomes are numbered according to length, with chromosome 1 being the longest and chromosome 22 being the shortest. A karyotype or karyogram is an ordered display of chromosomes. Some natural variation in relative chromosome length can be expected from person to person, so it is not always possible to distinguish each chromosome by its length. Therefore, the position of the centromere, which is the region of DNA responsible for movement of the replicated chromosomes into two daughter cells during mitosis and meiosis, also is used to classify chromosomes. The chromosomes in Figure 210 were stained with Giesma stain, resulting in distinctive chromosome bands. These form various patterns in the different chromosomes so that each chromosome can be distinguished easily. Using banding techniques, researchers can number chromosomes and study individual variations, missing or duplicated portions of chromosomes, which often result in serious disease, also are readily identified. More recently, techniques have been devised permitting each chromosome to be visualized with a different color. Chromosome abnormalities are the leading known cause of intellectual disability and miscarriage. Estimates indicate that a major chromosome aberration occurs in at least 1 in 12 conceptions. Most of these fetuses do not survive to term. About 50% of all recovered first trimester spontaneous abortuses have major chromosome aberrations. The number of live births affected by these abnormalities is, however, significant. Approximately 1 in 150 has a major diagnosable chromosome abnormality. Polyploidy cells with a multiple of the normal number of chromosomes are euploid cells. Because normal gametes are haploid and most normal somatic cells are diploid, these are both euploid forms. When a euploid cell has more than the diploid number of chromosomes, it is said to be polyploid cell. Several types of body tissues, including some liver, bronchial, and epithelial tissues are normally polyploid. A zygote has 3 copies of each chromosome rather than the usual 2. Has a form of polyploidy called triploidy. Nearly all triploid fetuses are spontaneously aborted or stillborn. The prevalence of triploidy among live births is approximately 1 in 10,000. Tetraploidy, a condition in which euploid cells have 92 chromosomes, has been found primarily in early abortuses, although occasionally affected infants have been born alive. Like triploid infants, however, they do not survive. Triploidy and tetraploidy are relatively common conditions, accounting for approximately 10% of all known miscarriages. Aneuploidy, spelled Aneuploidy. A cell that does not contain a multiple of 23 chromosomes is an aneuploid cell. A cell containing 3 copies of one chromosome is said to be trisomic, a condition termed trisomy, and is aneuploid. Monosomy, the presence of only one copy of a given chromosome in a diploid cell, is the other common form of aneuploidy. Among the autosomes, monosomy of any chromosome is lethal, But newborns with trisomy of chromosomes on 13, 18, 21, or X can survive. The difference illustrates an important principle. In general, loss of chromosome material has more serious consequences than duplication of chromosome material. Aneuploidy of the sex chromosomes is less serious than that of the autosomes. Very little genetic material, only about 40 genes, is located on the Y chromosome. For the X chromosome, inactivation of extra chromosomes largely diminish their effect. A zygote bearing no X chromosome, however, will not survive. A Aneuploidy is usually the result of non-disjunction, an error in which homologous chromosomes or sister chromosomes fail to separate normally. During mitosis or meiosis, non-disjunction produces some gametes that have two copies of a given chromosome and others that have no copies of the chromosome. When such gametes unite with normal haploid gametes, the resulting zygote is monosomic or trisomic. For that chromosome, occasionally a cell can be monosomic or trisomic for more than one chromosome. Autosomal aneuploidy. Trisomy can occur for any chromosome, but fetuses with other trisomies of chromosomes other than 13, 18, 21, or X do not survive to term. Trisomy 16, for example, is the most common trisomy among abortuses, but it is not seen in live births. Partial trisomy, in which only an extra portion of a chromosome is present in each cell, can also occur. The consequences of partial trisomies are not as severe as those of complete trisomies. Trisomies may occur in only some cells of the body. Individuals thus affected are said to be chromosomal mosaics, meaning that the body has two or more different cell lines, each of which has a different karyotype. Mosaics are often formed by early mitotic non-disjunction occurring in one embryonic cell but not the other. The best known example of aneuploidy is an autosome in trisomy of chromosome 21, which causes Down syndrome. Down syndrome is seen in approximately 1 in 800 to 1 in 1,000 live births. Its principal features are shown and outlined in Figure 2.13 and Table 2.1. The risk of having a child with Down syndrome increases greatly with maternal age. Women younger than 30 years of age have a risk ranging from 1 in 1,000 births to 1 in 2,000 births. The risks begin to rise substantially after age 35 and reaches 3 to 5% for women older than 45 years of age. This dramatic increase in risk is caused by the age of maternal egg cells, which are held in an arrested state of prophase 1 from the time they are formed in female embryo until they are shed in ovulation. Thus, an egg cell formed by 45-year-old women itself is 45 years old. The long suspended state causes defects to accumulate in the cellular proteins responsible for meiosis, leading to non disjunction. The risk of Down syndrome, as well as trisomies, does not increase with paternal age. The incidence of sex chromosome aneuploidies is fairly high. Among live births, about 1 in 500 males and 1 in 900 females have a form of sex chromosome aneuploidy because these conditions are genetically less severe than autosomal aneuploidies. All forms except complete absence of X chromosome material allow at least some individuals to survive. One of the most common sex chromosome aneuploidies affecting about 1 in 1,000 newborn females is trisomy X. Instead of 2 X chromosomes, these females have 3 X chromosomes in each cell. Most of these females have no overt physical abnormalities, although sterility, menstrual irregularity, or intellectual disability is sometimes seen. Some females have 4 chromosomes and they are more often intellectually disabled. Those with 5 or more X chromosomes generally have more severe intellectual disability and various physical defects. A condition that leads to somewhat more serious problems is the presence of a single X chromosome and no homologous X or Y chromosome, so that the individual has a total of 45 chromosomes. The karyotype is usually designated 45X, and it causes a set of symptoms known as Turner syndrome. Individuals with at least 2 X chromosomes and 1 Y chromosome in each cell karyotype have a disorder known as Klinefelter syndrome. In addition to the loss or gain of a whole chromosome, parts of chromosomes can be lost or duplicate, such as gametes are formed. And the arrangement of genes on chromosomes can be altered. Unlike aneuploidy and polyploidy, these changes sometimes have no serious consequence for an individual's health. Some of them can even remain entirely unnoticed, especially when very small pieces of chromosomes are involved. Nevertheless, abnormalities of chromosome structure can also produce serious disease in individuals or their offspring. During meiosis and mitosis, chromosomes usually maintain their structural integrity, but chromosome breakage occasionally occurs. Mechanisms exist to heal these breaks and usually repair them perfectly with no damage to the daughter cell. However, some breaks remain or heal in a way that alters the chromosome structure. The risk of chromosome breakage increases with exposure to harmful agents called clastogens. So this would be such as ionizing radiation, viral infections, and some types of chemicals. Deletions, broken chromosomes, and lost DNA cause deletions. Usually a gamete with a deletion unites with a normal gamete to form a zygote. The zygote thus has 1 chromosome with the normal complement of genes and 1 with some missing genes. Because many genes can be lost in deletion, serious consequences result, even though one normal chromosome is present. The most often cited example of a disease caused by a chromosomal deletion is the cry-do-chat syndrome. The term literally means cry of the cat and describes the characteristic cry of the affected child. Other symptoms include low birth weight, severe intellectual disability, microcephaly, and heart defects. The disease is caused by a deletion or part of the short arm of chromosome 5. A deficiency of genetic material is more harmful than an excess, so duplications usually have less serious consequences than deletions. For example, a deletion of a region of chromosome 5 causes cry-do-chat syndrome, but a duplication of the same region causes intellectual disability but less serious physical defects. Inversions. An inversion occurs when two breaks take place on a chromosome, followed by the reinsertion of the missing fragment at its original site, but in inverted order. Therefore, a chromosome symbolized as A, B, C, D, E, F, G might become A, B, E, D, C, F, G after inversion. Unlike deletions and duplications, no loss or gain of genetic material occurs, so inversions are balanced alteration of chromosome structure. And they often have no apparent physical effects. Some genes are influenced by neighboring genes, however, and this position affects a change in a gene's expression caused by its position sometimes results in a physical defect in these persons. Inversions can cause serious problems in the offspring of individuals carrying the inversion because the inversion can lead to duplications and deletions in the chromosomes transmitted to the offspring. The interchange of genetic material between non-homologous chromosomes is called translocation. A reciprocal translocation occurs when breaks take place in two different chromosomes and the material is exchanged. As with inversions, the carrier of reciprocal translocation is usually normal, but his or her offspring can have duplication and deletions. A second and clinically more important type of translocation is Robertson translocation. In this disorder, the long arms of two non-homologous chromosomes fuse at the centromere, forming a single chromosome. Robertsonian translocations are confined to chromosomes 13, 14, 15, 21, and 22 because the short arm of these chromosomes are very small and contain no essential genetic material. The short arms are usually lost during subsequent cell divisions. Because the carriers of Robertsonian translocations lose no important genetic material, they are unaffected, although they have only 45 chromosomes in each cell. Their offspring, however, may have serious monosomies or trisomies. For example, a common Robertsonian translocation involves the fusion of the long arms of chromosome 21 and 14. An offspring who inherits a gamete carrying the fused chromosome can receive an extra copy of the long arm chromosome 21 and develop Down syndrome. Robertsonian translocations are responsible for approximately 3 to 5% of Down syndrome cases. Parents who carry Robertsonian translocation involving chromosome 21 have an increased risk for producing multiple offspring with Down syndrome. Fragile sites. A number of areas on chromosomes develop distinctive breaks and gaps, observable microscopically. When the cells are cultured, most of these fragile sites do not appear to be related to disease. However, one fragile site located on the long arm of the X chromosome is associated with Fragile X syndrome. The most important feature of this syndrome is intellectual disability, with a relatively high population prevalence, affecting approximately 1 in 4,000 males and 1 in 8,000 females. Fragile X syndrome is the second most common genetic cause of intellectual disability after Down syndrome. In Fragile X syndrome, females who inherit the mutation do not necessarily express the disease condition, but they can pass it on to descendants who do express it. Ordinarily, a male who inherits a disease gene on the X chromosome expresses the condition because he has only one X chromosome. An uncommon feature of this disease is that about 1/3 of carrier females are affected, although less severely than males. Unaffected transmitting males have been shown to have more than about 50% repeated DNA sequences near the beginning of the Fragile X gene. These trinucleotide sequences, which consist of CGG sequences duplicated many times, cause Fragile X syndrome when the number of copies exceeds 200. The number of these repeats can increase from generation to generation. More than 20 other genetic diseases, including Huntington disease and myotonic dystrophy, are also caused by this mechanism. So, quiz check. What is the major composition of DNA? Define the terms mutation, autosomes, and sex chromosomes. What is the significance of mRNA? What is the significance of chromosomal translocation? Elements of formal genetics. The mechanisms by which an individual set of paired chromosomes produces traits are the principles of genetic inheritance. Mendel's work with garden peas first defined these principles. Later, geneticists refined Mendel's work to explain patterns of inheritance for traits and disease that appear in families. Analysis of traits that occur with defined, predictable patterns has helped geneticists geneticists assemble pieces of the human gene map. Current research focuses on determining the RNA or protein products of each gene and understanding the way they contribute to disease. Eventually, diseases and defects caused by single genes can be traced, and therapies to prevent and treat such diseases can be developed. Traits caused by single genes, called Mendelian traits. Each gene occupies a position along a chromosome known as a locus. The genes at a particular locus can have different forms called alleles. A locus that has two or more alleles that occur with each A locus that has two or more alleles that occur with an appreciable frequency in population is said to be polymorphic or to have polymorphism. Because humans are diploid organisms, each chromosome is represented twice, with one member of the chromosome pair contributed by the father and one by the mother. At a given locus, an individual has one allele whose origin is paternal and one whose origin is maternal. When the two alleles are identical, the individual is homozygous at that locus. When the alleles are not identical, the individual is heterozygous at that locus. Phenotype and genotype. The composition of genes at a given locus is known as the genotype. The outward appearance of an individual, which is the result of both genotype and environment, is the phenotype. For example, an infant who is born with an inability to metabolize the amino acid phenylalanine has the single gene disorder known as PKU, and thus has the PKU genotype. If the condition is left untreated, abnormal metabolites of phenylalanine will begin to accumulate in the infant's brain. An irreversible intellectual disability will occur. Intellectual disability is thus one aspect of the PKU phenotype. By imposing dietary restrictions to exclude food that contains phenylalanine, however, intellectual disability can be prevented. Foods high in phenylalanine include proteins found in milk, dairy products, meat, fish, chicken, eggs, beans, and nuts. Although the child still has PKU genotype, a modification of the environment produces an outwardly normal phenotype. This is why we do PKU testing in newborns. And FYI, the diet for these patients is super, super expensive. I have a friend with identical twins that have PKU, and it is outrageous how much it costs to feed them. Dominance and recessiveness. In many loci, the effects of one allele mask those of another when the two are found together in heterozygote. The allele whose effects are observable is said to be dominant. The allele whose effects are hidden is said to be recessive. Traditionally, for loci having two alleles, the dominant allele is denoted by an uppercase letter, and the recessive allele is denoted by a lowercase letter. When one allele is dominant over another, the heterozygote genotype as has the same phenotype as the dominant homozygote AA. For the recessive allele to be expressed, it must exist in the homozygote form AA. So large AA, small AA. A carrier is an individual who has a disease gene but is phenotypically normal. Many genes for a recessive disease occur in heterozygotes who carry one copy of the gene but do not express the gene. When recessive genes are lethal in the homozygous state, they are eliminated from the population when they occur in homozygotes by hiding in carriers. However, recessive genes for diseases are passed on to the next generation. Transmission of genetic diseases. The pattern in which genetic disease is inherited through generations is termed mode of inheritance. Knowing the mode of inheritance can reveal much about the disease-causing gene itself. And members of families with the disease can be given reliable genetic counseling. The known single gene diseases can be classified into four major modes of inheritance: autosomal dominant, autosomal recessive, X-linked dominant, and X-linked recessive. The first two types involve genes known to occur on the 22 pairs of autosomes. The last two types occur on the X chromosome. Very few disease-causing genes occur on the Y chromosome. The pedigree chart summarizes family relationships and shows which members of a family are affected by a genetic disease. Generally, the pedigree begins with an individual in the family, the proband. This individual is usually the first person in the family diagnosed or seen in a clinic. Autosomal dominance inheritance. Diseases caused by autosomal dominant genes are rare, with the most common occurring in fewer than 1 in 500 individuals. Therefore, it is uncommon for two individuals who are both affected by the same autosomal dominant disease to produce offspring together. Affected offspring are usually produced by the union of a normal parent with an affected heterozygous parent. The affected parent can pass either a disease-causing allele or a normal allele to the next generation. On average, half of the children will be heterozygous and will express the disease, and half will be normal. Several important characteristics of this pedigree support the conclusion that the trait is caused by an autosomal dominant gene. The two sexes exhibit the trait in approximately equal proportions. Males and females are equally likely to transmit the trait to their offspring. So this makes sense as to why dominant diseases or genetic issues have a 50% chance of occurring in offspring. No generations are skipped. If an individual has the trait, one parent must also have it. If neither parent has the trait, none of the children will have it, with the exception of a few mutations discussed later. Affected heterozygous individuals transmit the trait to approximately half their children, and because gamete transmission is subject to chance fluctuations, all or none of the children of affected parents may have the trait. When large numbers of matings of this type are studied, however, the proportion of affected children closely approaches 1/2. Parents at risk for producing children with a genetic disease nearly always ask the question, what is the chance that our child will have the disease? The probability that an individual will develop a genetic disease is termed the recurrent risk. When one parent is affected by an autosomal dominant disease and is heterozygote, and the other is unaffected, the recurrence risk for each child is 1/2. An important principle is that each birth is an independent event, much like a coin toss. Thus, even though parents may have already had a child with the disease, their recurrent risk remains 1/2, even if they have produced several children, all affected or unaffected by the disease. The law of independence dictates that the probability their next child will have the disease is still 1/2. Parents' misunderstanding of the principles is a common problem encountered in genetic counseling. So, meaning many people think that if they already have 2 children with the disease and they have 4 children, the next 2 will not have it. But actually, the next 2 still have a 50% chance of having the disease. If a child is born with an autosomal dominant disease and there is no history of the disease in the family, the child is probably the product of a new mutation. The gene transmitted by one of the parents has thus undergone a mutation from a normal allele to a disease-causing allele. The alleles at this locus in most of the parent's other germ cells are still normal. In this situation, the recurrence risk for their parents' subsequent offspring is not greater than that of the general population. The offspring of the affected child, however, will have a recurrence risk of 1/2 because these diseases often reduce potential for reproduction. Many autosomal dominant diseases result from new mutations. So, if it's not a genetic expression that the parent already has, and somehow that gene mutated in that particular pregnancy, and that child has it, so that child goes on to have a 50% risk for their children. However, if the parent has another child, the risk is not elevated. It is a normal risk that anybody without that genetic expression would have. Delayed age at onset. One of the best known autosomal dominant diseases is Huntington disease, a neurologic disorder whose main features are progressive dementia and increasingly uncontrollable limb movements. A key feature of this disease is its delayed age of onset. Symptoms usually are not seen until 40 years of age or later. Thus, those who develop the disease often have born children before they become aware that they have the disease causing mutation. If the disease was present at birth, nearly all affected persons would die before reaching the reproductive age. And the occurrence of the disease-causing allele in the population would be much lower. An individual whose parent has the disease has a 50% chance of developing it during middle age. He or she is thus confronted with a torturous question: should I have children, knowing that there is a 50-50 chance that I may have this disease causing gene and will pass it to half of my children? A DNA test can now be used to determine whether an individual has inherited trinucleotide repeat mutation that causes Huntington disease. Penetrance and expressibility. The penetrance of a trait is the percentage of individuals with a specific genotype who also exhibit the expected phenotype. Incomplete penetrance means individuals who have the disease-causing genotype may not exhibit the disease phenotype at all, even though the genotype and the associated disease may be transmitted to the next generation. A pedigree illustrating the transmission of autosomal dominant mutation with incomplete penetrance is provided in figure 2.22. Retinoblastoma, the most common malignant eye tumor affecting children, typically exhibits incomplete penetrance. About 10% of the individuals who are obligate carriers of the disease-causing mutation, i.e., those who have an affected parent and affected children and therefore must themselves carry the mutation, do not have the disease. The penetrance of the disease-causing genotype is then said to be 90%. The gene responsible for retinoblastoma is a tumor suppressor gene. The normal function of its protein product is to regulate the cell cycle so cells do not divide uncontrollably. When the protein is altered because of a genetic mutation, its tumor suppressing capacity is lost and a tumor can form. Expressivity is the extent of variation in phenotype associated with a particular genotype. If the expressivity of a disease is variable, penetrance may be complete, but the severity of the disease can vary greatly. A good example of variable expressivity is in an autosomal dominant disease is neurofibromatosis type 1 or von Recklinghausen disease, as in retinoblastoma. The mutations that cause neurofibromatosis type 1 can occur in a tumor suppressor gene. The expression of this disease varies from a few harmless cafe au lait, light brown spots on the skin, to numerous neurofibromas, scoliosis, seizures, gliomas, neuromas, malignant peripheral nerve sheath tumors, hypertension, and learning disorders. Several factors cause variable expressivity. Genes at other loci sometimes modify the expression of a disease-causing gene. Environmental, i.e., non-genetic factors also can influence expression of a disease-causing gene. Finally, different mutations at a locus can cause variation in severity. For example, a mutation that alters only one amino acid of the factor VIII gene usually produces a mild form of hemophilia. Hemophilia A, whereas a stop codon, which is a premature termination of translation, usually produces a more severe form of this blood coagulation disorder. Epigenetics and genomic imprinting. Although this chapter focuses on DNA sequence variation and its consequences for disease, there is increasing evidence that the same DNA sequence can produce dramatically different phenotypes because of chemical modifications altering the expression of genes. These modifications are collectively termed epigenetic. An important example of such modification is DNA methylation, the attachment of a methyl group to a cytosine base followed by a guanine base in the DNA sequence. These sequences, which are common near many genes, are termed CPG islands. When the CPG islands located near a gene become heavily methylated, the gene is less likely to be transcribed into mRNA. In other words, the gene becomes transcriptionally inactive. One study showed that identical Monozygotic twins accumulate different methylation patterns in the DNA sequences of their somatic cells as they age, causing increasing numbers of phenotypic differences. Intriguingly, twins with more differences in their lifestyles, such as smoking versus non smoking, accumulated larger numbers of differences in their methylation patterns. The twins, despite having identical DNA sequences, become more and more different as a result of epigenetic changes, which in turn affect the expression of genes. Epigenic alteration of gene activity can have important disease consequences. For example, a major cause of one form of inherited colon cancer, termed hereditary non-polyposis colorectal cancer, is the methylation of a gene whose protein product repairs damaged DNA. When this gene becomes inactive, damaged DNA accumulates, eventually resulting in colon tumors. Epigenetic changes are also discussed in chapters 3, 11, and 12. Approximately 100 human genes are thought to be methylated differently depending on which parent transmits the gene. This epigenetic modification, characterized by methylation and other changes, is termed genomic imprinting. For each of these genes, one of the parents imprints the gene or inactivates it when it is transmitted to the offspring. An example is the insulin-like growth factor 2 gene, IGF-2, on chromosome 11, which is transmitted by both parents, but the copy inherited from the mother is normally methylated and inactivated or imprinted. Thus, only one copy of IGF-2 is active in normal individuals. However, the maternal imprint is occasionally lost, resulting in two active copies of IGF-2. This causes excess fetal growth and contributes to a condition known as Beckwith-Wiedemann syndrome. A second example of genomic imprinting is a deletion of part of the long arm of chromosome 15, which, when inherited from the father, causes the offspring to manifest a disease known as Prader-Willi syndrome, short stature, obesity, hypogonadism. When the same deletion is inherited from the mother, the offspring develop Angelman syndrome, intellectual disability, seizures, ataxic gait. The two different phenotypes reflect the fact that different genes are normally active in the maternally and paternally transmitted copies of this region of chromosome 15. Characteristics of pedigrees: autosomal recessive inheritance. Like autosomal dominant diseases, diseases caused by autosomal recessive genes are rare in population. Although there can be numerous carriers, cystic fibrosis is the most common lethal recessive gene disease in white children. It occurs in about 1 to 2,500 births. Approximately 1 in 25 whites carries a copy of a mutation that causes cystic fibrosis. Carriers are phenotypically unaffected. Some autosomal recessive diseases are characterized by delayed age of onset, incomplete penetrance, and variable expressivity. The gene responsible for cystic fibrosis encodes a chloride ion channel in some epithelial cells. Defective transport of chloride ions leads to a salt imbalance, which results in secretions of abnormally thick, dehydrated mucus. Some digestive organs, particularly the pancreas, become obstructed, causing malnutrition, and the lungs become clogged with mucus, making them highly susceptible to bacterial infections. Deaths from lung disease or heart failure occurs before age 40 years in about half of the individuals with cystic fibrosis. The important criteria for discerning autosomal recessive inheritance include the following. Males and females are affected in equal proportions. Consanguinity, which is marriage between related individuals, is sometimes present, especially in cases of rare recessive diseases. The disease may be seen in siblings of affected individuals, but usually not in their parents. On average, 1/4 of the offspring of carrier parents will be affected. Recurrence risks. In most cases of recessive disease, both of the parents of affected individuals are heterozygous carriers. On average, 1/4 of their offspring will be normal homozygotes, half will be phenotypically normal carrier heterozygotes, and 1/4 will be homozygotes with the disease. Thus, the recurrence risk for the offspring of carrier parents is 25%. However, in any given family, there are chance fluctuations. If two parents have a recessive disease, they each must be homozygous for the disease. Therefore, all their children also must be affected. This distinguishes recessive from dominant inheritance because two parents, both affected by a dominant gene, are nearly always both heterozygotes, and thus 1/4 of their children will be unaffected. Because carrier parents usually are unaware that they both carry the same recessive allele, they often produce an affected child before becoming aware of their condition. Carrier detection tests can identify heterozygotes by analyzing the DNA sequence to reveal a mutation. Some recessive diseases for which carrier detection tests are routinely used include PKU, sickle cell disease, cystic fibrosis, Tay-Sachs disease, hemochromatosis, and galactosemia. Consanguinity and inbreeding are related concepts. Consanguinity refers to the mating of two related individuals, and the offspring of such matings are said to be inbred. Consanguinity is sometimes an important characteristic of pedigrees for recessive diseases because relatives share a certain proportion of genes received from a common ancestor. The proportion of shared genes depends on the closeness of their biologic relationship. Consanguineous matings produce a significant increase in recessive disorders and are seen more often in pedigrees for rare recessive disorders. X-linked inheritance. Some genetic conditions are caused by mutations in genes located on the sex chromosomes, and this mode of inheritance is termed sex-linked. Only a few diseases are known to be inherited as X-linked dominant or Y chromosome traits. So only the more common X-linked recessive diseases are discussed here. Because females receive two X chromosomes, one from the father and one from the mother, they can be homozygous for a disease allele and given locus, homozygous for the normal allele at the locus, or heterozygous. Males having only one chromosome are hemizygous for genes on this chromosome. If a male inherits a recessive disease gene on the X chromosome, he will be affected by the disease because the Y chromosome does not carry a normal allele to counteract the effects of the disease gene. Because a single copy of an X-linked recessive gene will cause disease in a male, whereas two copies are required for disease expression in females, more males are affected by X-linked recessive diseases than are females. X inactivation. In the 1950s, Mary Lewin proposed that one X chromosome in the somatic cells of females is permanently inactivated, a process termed X inactivation. This proposal, the Lyon hypothesis, explains why most gene products coded by the X chromosome are present in equal amounts in males and females, even though males have only one X chromosome and females have two X chromosomes. This phenomenon is called dosage compensation. The inactivated X chromosomes are observable in many interphase cells as highly condensed intranuclear chromatin bodies termed Barr bodies. Normal females have one Barr body in each somatic cell, whereas normal males have no Barr bodies. X inactivation occurs very early in embryonic development, approximately 7 to 10 days after fertilization in each somatic cell. One of the two X chromosomes is inactivated in some cells. The inactivated X chromosome is the one controlled by the father. In other cells, it is the one contributed by the mother. Once the X chromosome has been inactivated in a cell, all the descendants of that cell have the same chromosome inactivated. Thus, inactivation is said to be random but fixed. Some Some individuals do not have the normal number of X chromosomes in their somatic cells. For example, males with Klinefelter syndrome typically have two X chromosomes and one Y chromosome. These males do have one bar body in each cell. Females whose cell nuclei have three X chromosomes have two bar bodies in each cell. And females whose cell nuclei have four X chromosomes have three bar bodies in each cell. Females with Turner syndrome have only one X chromosome and no bar bodies. Thus, the number of bar bodies is always one less than the number of X chromosomes in the cell. All but one X chromosomes are always inactivated. Persons with abnormal numbers of X chromosomes, such as those with Turner syndrome or Klinefelter syndrome, are not physically normal. This situation presents a puzzle because they presumably have only one active chromosome. The same individuals with normal numbers of chromosomes. This is probably because the distal tips of the short and long arms of the X chromosome, as well as several other regions on the chromosome arm, are not inactivated. Thus, X inactivation is also known to be incomplete. The inactivated X chromosome DNA is heavily methylated. Inactive X chromosomes can be at least partially reactivated in vitro by administering 5-azacitidine, a demethylating agent. Sex determination. The process of sexual differentiation in which the embryonic gonads become either testes or ovaries begins during the 6th week of gestation. A key principle of mammalian sex determination is that one copy of the Y chromosome is sufficient to initiate the process of gonad differentiation that produces a male fetus. The number of X chromosomes does not alter this process. For example, an individual with two X chromosomes and one Y chromosome in each cell is still phenotypically a male. Thus, the Y chromosome contains a gene that begins the process of male gonadal development. This gene, termed SRY for sex determining region on the Y, has been located on the short arm of the Y chromosome. This SRY gene lies just outside the pseudo-autosomal region, which pairs with the distal tip of the short arm of the X chromosome during meiosis and exchanges genetic material with it. Crossover. Just as autosomes do, the DNA sequences of these regions on the X and Y chromosomes are highly similar. The rest of the X and Y chromosomes, however, do not exchange material and are not similar in DNA sequence. Other genes that contribute to male differentiation are located on other chromosomes. Thus, SRY triggers the action of genes on other chromosomes. This concept is supported by the fact that the SRY protein product is similar to other proteins known to regulate gene expression. Occasionally, the crossover between X and Y occurs closer to the centromere than it should, placing the SRY gene on the X chromosome after crossover. This variation can result in offspring with apparently normal XX karyotype but a male phenotype. Such XX males are seen in about 1 in 20,000 live births and resemble males with Klinefelter syndrome. Conversely, it is possible to inherit a Y chromosome that has lost the SRY gene, the result of either crossover error or deletion of the gene. This situation produces an XY female. Such females have gonadal streaks rather than ovaries and have poorly developed secondary sex characteristics. So, quiz check. Why is the influence of the environment significant to phenotype? Discuss the differences between dominant allele and recessive allele. Why are the concepts of variable expressivity, incomplete penetrance, and delayed age of onset so important in relation to genetic diseases? What is the recurrence risk for autosomal dominant inheritance and recessive inheritance? Characteristics of pedigrees. X-linked pedigree shows distinctive modes of inheritance. The most striking characteristic is that females seldom are affected. To express an X-linked recessive trait fully, a female must be homozygous. Either both her parents are affected or her father is affected and her mother is a carrier. Such matings are rare. The following are important principles of X-linked recessive inheritance. The trait is seen much more often in males than in females because a father can give a son only a Y chromosome. The trait is never transmitted from father to son. The gene can be transmitted through a series of carrier females. The gene is passed from an affected father to all his daughters, who, as phenotypically normal carriers, transmit it to approximately half their sons who are affected. A relatively common X-linked recessive disorder is Duchenne muscular dystrophy, DMD, which affects approximately 1 in 3,500 males. As its name suggests, the disorder is characterized by progressive muscle degeneration. Affected individuals usually are unable to walk by age 10 or 12. The disease affects the heart and respiratory muscle, and death caused by respiratory or cardiac failure usually occurs before age 20. Identification of the disease-causing gene on the short arm of the X chromosome has greatly increased our understanding of the disorder. The DMD gene is the largest gene ever found in humans, spanning more than 2 million DNA bases. It encodes a previously undiscovered muscle protein termed dystrophin. Extensive study of dystrophin indicates that it plays an essential role in maintaining the structural integrity of muscle cells. It may also help regulate the activity of membrane proteins. When dystrophin is absent, as in DMD, the cell cannot survive and muscle deterioration ensues. Most cases of DMD are caused by frameshift deletions of portions of the DMD gene and thus involve alterations of the amino acid encoded by the DNA following deletion. Reoccurrence risks. The most common mating type involving X-linked recessive genes is the combination of a carrier female and a normal male. On average, the carrier mother will transmit the disease, causing allele to half her sons and half her daughters. So the sons are affected, the daughters are carriers. The other common mating type is affected father and a normal mother. In this situation, all the sons will be normal because the father can only transmit his Y chromosome to them. Because all the daughters must receive the father's X chromosome, they will all be heterozygous carriers. Because the sons must receive the Y chromosome and the daughters must receive the X chromosome, with the disease gene, they are precise outcomes and not probabilities. None of the children will be affected. The final mating pattern, less common than the other two, involves an affected father and a carrier mother. With this pattern, on average, half the daughters will be heterozygous carriers and half will be homozygous for the disease allele and thus affected. Half of the sons will be normal and half will be affected. Some X-linked recessive diseases such as DMD are fatal or incapacitating before affected individuals reaches reproductive age, and therefore affected fathers are rare. Sex-limited and sex-influenced traits. A sex-limited trait can only occur in one sex, often because of anatomic differences. Inherited uterine and testicular defects are two obvious examples. A sex-influenced trait occurs much more often in one sex than the other. For example, male pattern baldness occurs in both males and females, but is much more common in males. Autosomal dominant breast cancer, which is much more commonly expressed in females than males, is another example of a sex-influenced trait. Linkage analysis and gene mapping. Locating genes on a specific region of chromosomes has been one of the most important goals of human genetics. The location and identification of a gene can tell much about the function of the gene, the interaction of the gene with other genes, and the likelihood that certain individuals will develop a genetic disease. Classic pedigree analysis. During the first meiotic stage, the arms of homologous chromosome pairs intertwine and sometimes exchange portions of their DNA in a process known as crossover. During crossover, new combinations of alleles can be formed. For example, two loci on a chromosome have alleles A capital and alleles A lowercase, so dominant and recessive. And alleles B and B, same, dominant and recessive. Alleles A and B, dominant, are located together on one member of a chromosome pair, and alleles A and B recessive are located on another member. The genotype of this individual is denoted as AB/AOB, so dominant AB / recessive AOB. All four possible pairs of alleles can be transmitted to the offspring. The process of forming such new arrangements of alleles is called recombination. Loci that are located very close to one another are unlikely to experience recombination and are said to demonstrate linkage. The frequency of recombination can be assessed in families and is used to determine the relative positions of loci on chromosomes. Complete human gene map, prospects and benefits. The major goals of the Human Genome Project were to find locations of all human genes and to determine the entire human DNA sequence. These goals have now been accomplished, and the genes responsible for approximately 5,000 Mendelian conditions have been identified. This has greatly increased our understanding of the mechanisms that underlie many diseases, such as retinoblastoma, cystic fibrosis, neurofibrosis, and Huntington disease. The project also has led to more accurate diagnosis of these conditions and, in some cases, more effective treatment. DNA sequencing has become much less expensive and more efficient in recent years. Consequently, hundreds of thousands of individuals have now been sequenced, leading in some cases to the identification of disease-causing genes. Did you know? Thousands of subjects are currently enrolled in a gene therapy clinical trials, and several gene therapy treatments have been approved by the U.S. Food and Drug Administration. Most trials and treatments involve the genetic alteration of cells to combat various types of cancers. Others involve the treatment of inherited diseases such as B, thalassemia, hemophilia, severe combined immunodeficiency, and retinitis pigmentosa. We're on the last page. Multifactorial inheritance. Not all traits are produced by single genes. Some traits result from several genes acting together. These are called polygenic traits. When environmental factors influence the expression of the trait, as is usually the case, the term multifactorial inheritance is used. Many multifactorial and polygenetic traits tend to follow a normal distribution in populations. If both alleles at each of the three loci are white alleles, the color is pure white. If most alleles are white but a few are red, the color is somewhat darker. If all are red, the color is dark red. Other examples of multifactorial traits include height and intelligence quotient, IQ. Although both height and IQ are determined in part by genes, they are also influenced by environment. For example, the average height of many human populations has increased 5 to 10 centimeters in the past 100 years because of improvements in nutrition and healthcare. Also, IQ scores can be improved by exposing individuals, especially children, to enriched learning environments. Thus, both genes and environment contribute to the variation in these traits. A number of diseases do not follow the bell-shaped distribution. Instead, they appear to be either present in or absent from an individual. Yet they do not follow the patterns expected of single gene diseases. Many of these are probably polygenic or multifactorial, but a certain threshold of liability must be crossed before the disease is expressed. Below the threshold, the individual appears normal. Above it, the individual is affected by the disease. A good example of such threshold trait is pyloric stenosis, a disorder characterized by a narrowing or obstruction in the pylorus, the area between the stomach and small intestine. Chronic vomiting, constipation, weight loss, and electrolyte imbalance can result from the condition, but it is easily corrected with surgery. The prevalence of pyloric stenosis is about 3 in 1,000 live births in whites. This disorder is much more common in males than females, affecting 1 in 200 males and 1 in 1,000 females. The apparent reason for this difference is the threshold of liability is much lower in males than females. Thus, fewer defective alleles are required to generate the disorder in males. This situation also means the offspring of affected females are more likely to have pyloric stenosis because affected females necessarily carry more disease-causing alleles compared with most affected males. A number of other common diseases are thought to correspond to a threshold model. They include cleft lip and cleft palate, neural tube defects such as anencephaly and spina bifida, clubfoot, which is Talipes, and some forms of congenital heart disease. Although reoccurrence risk can be given with confidence for single gene diseases, such as 50% for autosomal dominant diseases and 25% for autosomal recessive diseases, it is considerably more difficult to do so for multifactorial diseases. The number of genes contributing to the disease is not known. The precise allele constitution of the biologic parents is not known, and the extent of environmental effects can vary from one population to another. for most multifactorial diseases. Empirical risks, i.e., those based on direct observation, have been derived. To determine empirical risks, a large sample of biological families in which one child has developed the disease is examined. The siblings of each child are then surveyed to calculate the percentage who also develop the disease. Another difficulty is distinguishing polygenic or multifactorial diseases from single gene diseases having incomplete penetrance or variable expressivity. Large data sets and good epidemiologic data are often necessary to make the distinction. The genetics of common disorders such as hypertension, heart disease, and diabetes is complex and often confusing. Nevertheless, the public health impact of these diseases, together with the evidence for hereditary factors in their etiology, demands that genetic studies be pursued. Thousands of genes contributing to susceptibility for these diseases have been discovered, and the next decade will undoubtedly witness substantial advancements in our understanding of these disorders. Quick check. Define linkage analysis. Cite an example. Why is threshold of liability an important consideration in multifactorial inheritance? Discuss the concept of multifactorial inheritance and include 2 examples. So we're going to read box 2.1 real quick. This is the criteria used to define multifactorial diseases. The recurrence risk becomes higher if more than one family member is affected. For example, the recurrence risk for neural tube defects in a family increases by 10% if 2 siblings have been born with the disease. By contrast, the recurrence risk for single gene diseases remains the same regardless of the number of siblings affected. If the expression of the disease is more severe, the recurrence risk is higher. This is consistent with the liability model. A more severe expression indicates that the individual is at the extreme end of the liability distribution. Relatives of the affected individual are thus at a higher risk for inheriting disease genes. Cleft lip or cleft palate is a condition in which this has been shown to be true. Relatives of probands of the less commonly affected are more likely to develop the disease, as with pyloric stenosis. This occurs because an affected individual of the less susceptible sex is usually at a more extreme position on the liability distribution. Generally, if the population frequency of the disease is female, the risk for offspring and siblings of probands is appropriately I think that means squared. This does not usually hold true for single gene traits. The recurrence risk for the disease decreases rapidly in more remotely related relatives. Although the recurrence risk for single gene diseases decreases by 50% With each degree of relationship, e.g., an autosomal dominant disease has a 50% recurrence risk for siblings, 25% for uncle-nephew relationship, 12.5% for first cousins. The risk for multifactorial inheritance decreases much more quickly. And that is the end of Chapter 2, Genes and Genetic Diseases. Good luck on your test.